The other technique, 2-deoxyglucose, this is basically where we inject a critter with glucose that has a little radioactive molecule attached to it. And whenever neurons are activated in the brain, they consume glucose as fuel. So the more activated a neuron is, the more radioactive glucose it's going to have in it, and you can then measure the radioactive glucose in the brain. When you do this, when you use this technique, and you use it in combination with behavioral testing, so you look not only at the brain activity of the animal when it's exposed to chemicals, but you also look at the behavior. For example, teaching an animal to fear a certain smell or not fear, or to like a certain smell. What we find is that there's a pattern of neural activation that's related to both the chemical structure and to the perception. So we're able to map these patterns of activation that seem to match up with what the animal seems to be perceiving, as well as the chemical structure of the compound that's being perceived. So that's kind of cool, right? Where we think that our ability to detect all of our different smells is really based on the activation, the way that the olfactory receptors, the pattern of activation of olfactory receptors, and the pattern of activation of the glomeruli. So here's an example of some data from the 2-deoxyglucose studies. This is kind of like a little heat map, like one of those weather radar things where if you see like red and orange, there's like lots and lots of activity, lots of storms, and where you see blue, there's no activity, and green is a little activity. It's the same here. So we have these different smells going all the way from alpha-phelandrine all the way to valeric acid, and notice that each one of these different compounds produces a distinct pattern of activation in the olfactory bulb, to where you can tell if you were to look at one of these, you could tell which chemical the animal was smelling based on the pattern of activation that you see here. So the signals from the olfactory bulb are sent to, first of all, the primary olfactory cortex that's located in the temporal lobe and the amygdala. Now, the amygdala connection is really important because the amygdala is one of those structures in the brain that is very strongly implicated in our emotional perception, our emotional tone. In other words, the coloring of our experiences with emotion. The amygdala seems to play a huge role in that. And think about all the times you've smelled something that's pleasant and it's relaxed you, like lavender, right? Lavender is supposed to relax people. And then think of all the times you've smelled something that's really unpleasant and it's kind of made you real tense, like gasoline or chemicals or other types of nasty smells. And you'll see that the reason for this is because there's a very direct connection between our perceptions, our olfactory bulbs, and the parts of our brain that mediate our emotional state. Then from there, they're processed there, and then on to the secondary olfactory, the orbitofrontal cortex and the frontal lobe, which is like right here, basically right on top of your eyeballs is the orbital frontal cortex. That's a place that does further processing of the smells that you're smelling. So here we have a cartoon drawing. This is a picture of the bottom of the brain. So if I took my brain out, you're looking at the bottom of it. That's what this is. This would be the front right here. This is the back of your head here. Front of your head, back of your head. This is the bottom, so your eyeballs would be right about here. So underneath the brain you have the olfactory bulbs, which are these two little thin structures here. Information comes from your nose, goes into the olfactory bulb, from the olfactory bulb to the piriform cortex, and also to the amygdala, to the temporal lobe, and then finally to the orbitofrontal cortex right about here. And here's a nice cartoon drawing of the relationship between all of these structures. Notice that from the olfactory mucosa, we go from the olfactory bulb to the piriform cortex to the orbital frontal cortex, and notice how these inputs all feed to the amygdala. So all these systems are processing smell information and sending it all to one of the parts of our brain that mediates emotional states, and that is the amygdala. In the piriform cortex, the first stop in the cortex, we have experiments by Renneker that are mentioned in your book, where Renneker used multiple microelectrodes to measure individual neurons in the piriform cortex, and found that a smell from isoamyl acetate causes activation across the cortex. So he was able to identify the specific neurons that fired, this pattern of neurons that fired as a response to isoamyl acetate. Settler and Axel used optical imaging and observed the same pattern. So instead of individual recordings, what they did was used this red light, this lighting techniques to see which neurons lit up as a result of being stimulated with isoamyl acetate, and they got the same the results as Renneker did. So here are the data, right? So here are the patterns of activity in the piriform cortex from electrode placements, and the lighter the square, the more activity. And then notice here we have the activation by light activation by isoamyl acetate. Notice very similar patterns of responding. Notice that you have a lot of responding here on the bottom part, and most of the responding here is confined to the bottom part here. So there's a lot of responding, a lot of correlation between these two types of studies. Here we have activity throughout the piriform cortex, and we're looking at the pattern responding to two different chemicals, hexanol and octanol. The hexanol is the pattern of responses that you see with green, and the octanol is the pattern responses that you see in red. One of the things you want to notice here is that there's a strong distinction between these two chemicals. In other words, red is red. The cells that are stained in red here in this image only respond to octanol, whereas the cells that are stained in green only respond to the hexanol. Really interesting that we're able to see all the way starting from the glomeruli, even from the receptors. We're seeing that there's this pattern, that the coding of smell seems to be this pattern as we go higher and higher in the brain. So once you smell something, how is that thing represented in your brain? How is an odor object represented? Where is it represented? How is it represented? Well, Wilson did an experiment where he measured the response of neurons in the rat's piriform cortex to two different odorants. And basically what we have was isoamyl acetate mixed with peppermint. Remember, peppermint is very strong. And then just isoamyl acetate alone. So we have this mixture of isoamyl acetate and peppermint, and then just the acetate alone. And what we found was that if you expose the animal to both the mixture and the compound, eventually you could see differences in the firing patterns in the piriform cortex between the isoamyl acetate and the mixture of the acetate and the peppermint. Now this is really cool because this is from experience. What we're seeing is this training of trying to tell the difference between these two. And when the animal is able to perceive the difference, we are able to see this difference in the piriform cortex. We're able to see that they light up different patterns of activation in the piriform cortex. So here we have these areas of the cortex that receive input from the olfactory bulb. And what happens is eventually some pattern is formed. So that's represented here in B by these little areas being linked together. And then later on, so this would be, for example, the isoamyl acetate, and then eventually we would see the isoamyl acetate and the peppermint smell light up this particular pattern here. So these individual patterns are going to light up in certain ways together at certain times to represent different odors. So here's another example. So we have a flower that emits all these molecules. In the olfactory bulb, this map of particular receptor neurons is activated. This then sends activation to the piriform cortex. And at first you see this scattered activation where it just lights up a certain pattern. But after a while of exposure to these odorant molecules, you start to see that all of these different areas in the piriform cortex work together, light up together to represent that particular thing that you're smelling. So what we're thinking here, the hypothesis is that our ability to recognize different objects, the smells of different things, is based upon a learned pattern of activation, probably starting in the piriform cortex. So that's smell. Let's talk about flavor now. We talked about taste. Flavor is a mixture of smell and taste and other sensations, right? So if you've ever tried to eat something when you've had a really bad cold or congestion, you'll notice that things don't taste the same. That's because a lot of our taste, the odor stimuli from the food, it does reach up to the olfactory mucosa through the retronasal route, so that in the back of your throat you have a little passage that goes up up into your nose. If you've ever blown milk or puke or something out of your nose, you know this, right? You know there's a connection there. And so whenever you eat something, the smell of that does go up into your nose, so it adds to the taste experience. So the taste of most compounds, the taste of most things that you eat is influenced by olfaction, but a few aren't. For example, the chemical MSG, monosodium glutamate, which is added to a lot of foods as a flavor enhancer, it doesn't depend on smell. It's entirely based on the activation of molecules on your tongue. So here's a cutaway drawing of a human being, right? So here we have the mouth and the tongue. So the food goes in here, but of course, as I said, there is this connection between your oral cavity and your nasal cavity. So whenever you eat, anything you eat here, the smell, the chemicals, some of the smell does make its way easily up into here, and that adds to the perception of flavor.